Hello, everyone. Welcome to The Current. This is the North Central Region Water Network's Speed Networking webinar series. I'm Rebecca Power. I'm joining you from the University of Wisconsin, and I will be moderating today's session. For those of you that are not familiar with the North Central Region Water Network, we are a collaboration among land-grant universities and partners in 12 upper Midwestern states, and we are led by our university extension systems. So thanks for joining us today. Our topic, as you can see on the screen, is emerging contaminants, the latest research on PFAS, which I'm sure many of you know by now are perfluoroalkyl and polyfluoroalkyl substances, man-made chemicals, a large number of man-made chemicals that have been used in common household and industrial products, and whether you have a professional interest in this topic and are working on it professionally or are impacted by PFAS personally, this is obviously a critical topic, and we are happy to bring you some really talented professionals here in our region that are working on this issue. So we're going to be covering some of the latest research on PFAS, including impacts on reproductive and child health, the biological effects of exposure on fish and wildlife populations, and we'll also give you kind of a PFAS 101 right at the beginning. So the way that these sessions work, we will ask for your questions for presenters in the chat box. That chat box is accessible through the purple, what we call the collaborate panel in the lower right-hand corner of your screen. There's a little chat bubble there that you can click on to get your questions in there. And we will save those questions until all of our presenters are through. Normally these sessions are, we'll have 3 presenters, 10 minutes each is our typical format. Today, Courtney is going to go a little bit longer and give us some of that PFAS 101, but we'll try to save as much time for questions at the end as we can, while taking enough time to cover the details of this topic. If you're having audio issues, there is a phone option that can be accessed through the session menu, which is in the upper left-hand corner of your webinar screen, and selecting use your phone for audio. And just a reminder for folks to keep themselves muted if you're not speaking. Okay, so today's presenters, just a very quick overview, and we'll introduce them in more detail in a minute. Courtney Carignan, assistant professor and food science, human nutrition, and pharmacology and toxicology expert at Michigan State University. Masa Modiri Garavaran, postdoctoral research assistant at Purdue. And Cheryl Murphy, associate professor of ecotoxicology of fish at Michigan State University. And Courtney is brave enough to be joining us. She's got a stomach bug that had struck her down, but is not keeping her from this webinar today. So thank you, Courtney, for joining us. And here on the screen, I'm not going to read this for you, but it's a little bit about Dr. Carignan and the experience that she brings to this topic. And with that, we will get right on to her presentation. So thank you so much, Courtney.
Yeah, thank you for having me. Can you hear me okay?
Yes, you sound good, and just make sure to speak up.
All right, great. Poly and perfluoroalkyl substances, or PFAS, are added to products to impart stain and water repellency, including for some types of shoes, upholstery, carpeting, clothing, paper, food packaging, nonstick cookware, and AFFF, aqueous film-forming foams, which are used to fight fuel fires. While convenient, they're associated with serious health problems, and they can remain in the environment forever. They're man-made, manufactured, put into products, and disposed of. They can make their way from industrial plants and landfills into waterways, and then into our food and drinking water. We're also exposed to them from products in our homes and our workplaces. Most people across the US and the world have measurable levels of PFAS in their bodies. Once in the body, they bind tightly to proteins in the blood, where they can recirculate and persist. Half of the amount that enters your body today could remain with you for years to come. We also know that they pass easily into the womb, exposing the fetus, which is especially vulnerable. PFOA and PFOS, called C8 because they have 8 carbons, were phased out of use a decade ago due to concerns about their toxicity and persistence. They're characterized by a chain of carbon-fluorine bonds that are incredibly strong and is what makes them so persistent. One end of the chain is attracted to water and the other repels it, a unique characteristic that makes them useful, but also enables them to be highly mobile in the environment. PFOA and PFOS have been linked to a number of health concerns. One of the largest and most well-known health studies is from the Mid-Ohio River Valley, where PFAS discharge from industry contaminated 5 water districts. The health study included over 69,000 participants who provided blood samples and access to their medical records. 3 scientists were tasked with determining, based on the weight of evidence, whether given health effects were more probable than not to have been caused by this exposure. The science panel agreed on the 6 health effects that are listed here, and I will review them very briefly. PFOA levels in the blood were positively associated with higher measurements of cholesterol, a 16% increased odds on having above normal levels of ALT, which is a marker for hepatocellular injury. Effects on liver function are supported by other human and animal studies. PFOA levels in the blood were positively associated with higher measurements of uric acid, which is an indicator of kidney function and is used to diagnose gout. Similar associations have been observed for about 10 other populations, including the general public, children, and workers. 2 meta-analyses now have reported that positive associations with thyroid-stimulating hormone are most consistent for pregnant women and teenage boys, and that overall findings suggest that they can induce thyroid dysfunction and disease. Evidence of carcinogenicity date back to rodent and worker studies published as early as the 1980s and 90s, indicating links to testicular, prostate, bladder, and kidney cancers. Increased odds of several cancers were observed among those with drinking water exposure in the Mid-Ohio River Valley, particularly for kidney and testicular cancer, which both had a doubling of odds. I'd like to do a thought exercise to provide some context about what that means. Recall that personal odds are impacted by many factors, so here we're discussing population risk. If the lifetime risk for developing kidney cancer is about 1 in 63, or about 1.6%, a doubling of risk is about 1 in 32, or about 3.1%. So in a population for 69,000, that's approximately an additional 1,000 cases of kidney cancer. And among 6 million people, which I'll come back to in a minute, that's an additional 95,000 cases. The point here is that seemingly small increased odds can have a big effect at a population level. IARC now classifies PFOA and PFOS as human carcinogens based on both animal and human studies. And a medical monitoring program has been recommended for people in the Mid-Ohio River Valley that includes screening for high cholesterol, thyroid disease, testicular and kidney cancers, and ulcerative colitis. Ulcerative colitis is an autoimmune disease, which brings me to effects of these chemicals on immune function. A 2016 systematic review by the National Toxicology Program concluded that PFOA and PFOS are hazardous to the immune system. This finding was based on human and animal studies reporting that PFOA and PFOS cause reduced resistance to infectious disease, increased hypersensitivity-related outcomes, and increased incidence of autoimmune disease. Our current understanding is that it appears to inhibit the body's ability to mount a response to a pathogen, which is the adaptive immune system, but to program the innate immune system to react inappropriately, such as in the case of autoimmune disease. Some PFASs have been shown to interfere with proper functioning of thyroid and sex hormones, which are important for fertility, healthy pregnancy, and fetal development. A handful of studies have reported increased exposure to be associated with increased odds of infertility, lower sperm concentration and count, fewer normal sperm, and increased post-implantation loss or miscarriage. Many studies have investigated developmental effects in both animals and humans, and a systematic review concluded that developmental exposure to PFOA can impact fetal growth. Another developmental endpoint affected by some PFAS is on the mammary glands, with structural changes that persist into adulthood. This has potential implications for breast cancer, as well as a woman's ability to lactate. Another important point to mention is that both breast milk and drinking water that contain PFAS contribute to increased exposure for infants, which is also a sensitive developmental window, and highlights the need to reduce exposure at the population level. Last year, ATSDR, which is part of the CDC, released an 800-page report reviewing existing health literature for PFAS and identified the immune and developmental effects to be the most sensitive endpoints for PFOA and PFOS, which means that they occur at the lowest levels of exposure. Extrapolation of these levels support drinking water health advisories below 70 parts per trillion. States have begun passing their own regulatory limits that are both lower than the EPA health advisory and enforceable. Michigan has proposed some of the lowest, which I've included at the end of this slide deck. We've talked a lot about PFOA and PFOS, which are now considered legacy PFASs and were phased out of use over a decade ago. However, hundreds of replacements are now in use that include several subclasses of PFAS. Less is known about the safety of these chemical cousins, and traditional filtration methods like activated carbon have been shown to be less effective at removing them from contaminated water. PFAS are unlike other persistent organic pollutants in that they easily migrate into surface and groundwater. However, like other POPs, they build up in the body over time, such that even relatively low drinking water concentrations can substantially increase human body burden. Levels above EPA's health advisory of 70 parts per trillion can increase serum levels more than 4 times that of background. A few years ago, I helped publish a study reporting that over 6 million Americans have been served by drinking water systems containing elevated levels of PFAS. This was a first look based on limited data, and it's since been estimated that PFAS are detectable in drinking water for 110 million Americans. You'll notice on this map that Michigan really lights up. That's because Michigan recently undertook one of the most comprehensive surveys, testing over 1,700 public water systems that serve 80% of state residents. PFAS was detected in approximately 170, or about 10% of those systems, with levels above 10 parts per trillion, about 51 of them, which is around 3%. Point sources of contamination include military bases and airports, tannery operations, paper mills, and plating facilities. Access to clean drinking water is a common challenge for impacted communities. Short-term solutions include distribution of bottled water and installation of home filtration systems, whereas longer-term solutions include filtration systems for city water or connection to clean water supplies. PFAS-contaminated groundwater can discharge into water bodies. An unusual foam has been discovered now at several contaminated sites. The foam is whiter and more airy than common lake foam and is tested for high levels of PFAS. People from one community say that their children used to play in the foam, building snowmen and making fake beards. There are now advisories not to eat or touch the foam due to potential health concerns. That same community has a do not eat advisory for deer. And many impacted communities have do not eat advisories for local fish, as well as foods that were grown or produced using contaminated water. We know that PFAS can be taken up into some plants, and that the extent of uptake is generally mediated by the chain link. One of the most common questions from impacted communities is about consumption of homegrown and produced foods. To help address these questions, I'm part of a team that will be sampling foods from PFAS-impacted areas across the country and estimating exposure from both drinking water and diet in order to improve our understanding and inform future recommendations. Exposed communities also have many questions related to exposure and health impacts of PFAS. To help address these, I'm working with a national health study led by the CDC and ATSDR that is modeled off the health study and includes several of the communities I work with in Michigan and the Northeast. I'm also helping lead a project to investigate effects of AFFF contaminated drinking water on children's immune systems. Other parts of the project include developing an online resource center for impacted communities and interviewing people to understand their exposure experience. Twice now, we've organized national conferences on PFAS at Northeastern University in Boston, designed to help support impacted communities across the nation. And I'm leading something similar at the state level here in Michigan. Organizations tend to be insular, but complex problems require multidiscipline and multi-institution teams. I greatly appreciate the chance to speak with you all today, and I hope that sharing this information will help support you to take measures to protect environmental public health in your state. Thank you.
Amazing. Thank you, Courtney, so much. One of the purposes, in addition to presenting this information to everyone, one of the things we do or want to do with these webinars is introduce you to the people around the region that are doing this work that you may not have had the chance to meet yet, so to speak. So thank you, Courtney, so much for sharing the information a little bit about your work. Okay, and we're just going to skip over these some of these extra slides that Courtney had and go on to Masa Mudiri Garavaran. And Masa, as I said, she is at Purdue University. She's a postdoctoral research person there, and she's going to talk to us a little bit about the occurrence of PFAS in compost and biosolids and remediation approaches. So it's a little bit about her on this introductory slide. And we'll go ahead and get started with her presentation. So thank you, Masa.
Everyone, joining us today. Today, I talk about the occurrence of these compounds in compost and biosolids and some remediation approaches. I work on the guidance of Dr. Lindali at Purdue University, and we are doing research on PFAS compounds. So let's see, after the brief introduction, sorry, I hit that twice. So after the brief introduction that Courtney gave us, I would like to give more details about the nature of these compounds because that's necessary to be able to treat them in the environment. And so there are different ways that these compounds can actually introduce into the environment. And today I will mostly focus on the biosolids and the occurrence of PFAS in biosolids that are mostly land applied, and also the contamination that happens in groundwater, which is a result of using most of these firefighting foams in military bases, sites, in airports, and other sources. Given this information, I would like to give a little bit more closer look to these compounds. So PFAS compounds can either be like PFAS precursors, which means that these compounds are very persistent and they won't go away. So if these PFAS compounds are breaking down, it is possible that they are breaking to some other subclass of PFAS compounds, which are well known as perfluoroalkyl acids. These perfluoroalkyl acids are really persistent. These are anionic, they have low pKa, and it makes them more soluble and more mobile in our water sources. And these alkyl acids can be either having a carboxylate attached to them or a sulfonate group attached to them, which will make a huge difference in their behavior. Also, these are termed as long chain and short chain depending on their different structures and the perfluoroalkyl chain that these compounds have. So given this, let's see why we use these organic waste-based soil amendments in our environment. The first benefit is actually the sustainability, because by using these waste-based products, we are able to recycle waste for plant nutrients and also improve soil health, which can decrease the waste and sometimes the landfills. And the challenge is that what if the PFAS can reach from these potential sources to our water sources? And if the PFAS are present, what kind of PFAS are present in our products? At Purdue, we have been quantifying the PFAS concentrations in different types of waste route and commercially available organic products. So let's see what's the PFAS fate in water resource and recovery facilities. Unfortunately, after PFAS is introduced to wastewater influent through different sources, it's not going away. It's not treated in wastewater treatment. So it can end up in the sludge, these compounds can sorb to a sludge and just get accumulated over time. And at the end of the day, they will either end up in effluent discharge or in the biosolids. But the problem is that there is some evidence that the concentrations of these PFAS can be higher in biosolids compared to whatever we have introduced to wastewater treatment plant. And that is because we have some precursors present in sludge or biosolids that can degrade to PFAS and increase their final concentrations. Given this information, let's see what can be the occurrence happens. So we looked at the commercially available organic products and also the organic fraction of municipal organic solid waste. And we looked at to see what kind of PFAS or PFAS are present in these commercially available waste-derived products. So in this slide, we actually see a brief introduction to the products that we have looked at through. So we looked at some commercially available products that some of them were biosolid based and some of them were not biosolid based. And for municipal organic solid waste, we got some samples that they had the compostable food packaging in them, and some of them they did not have. And given this information, when we looked at the PFAS concentration, we found a higher concentration of PFAS compounds in biosolid-based products compared to non-biosolid-based products. And we found that longer chains or more perfluoroalkyl, higher perfluoroalkyl chains were dominant. And also we did extra screening with some advanced high-resolution mass spectrometry, which we found some precursors that could degrade to these PFAS compounds. And when we looked at the compost from the municipal solid waste, we found that there is higher PFAS concentrations in the compost that had food packaging inside of them. And for the rest of them, they were lower concentrations. And the data we found here led to Washington's Healthy Food Packaging Act. And for second part of my talk, I will talk about the contamination which has happened in groundwater. Unfortunately, substantial soil and groundwater contamination has been observed in the vicinity of fire training areas, which brings the need for an effective inside technology that can treat these compounds. And that's why we chose an iron-based bimetal technique that has potential to be used in situ, which means that we can use it as permeable reactive barrier, which possibly can intercept PFAS contaminated groundwater. The whole idea is that if we have a contaminated groundwater with PFAS, can we pass this contaminated water through the permeable reactive barrier, which is filled with our techniques particles? Can this water be treated after passing through this barrier? And then, so let's see what this technique is. So basically, we use nanoparticles and we synthesize them onto activated carbon. And the benefit that this technique has is this technique is able to transform PFAS that are even resistant to chemical oxidations. But there are some questions that need to be answered, such as what are the major products, or does the structure of PFAA affect this treatment? What is the effect of temperature? And to answer these questions, we've been conducting some batch and column experiments to see the efficiency of this technique at Purdue University. And when we looked at the C8 or PFOS transformation with this technique, we found that during the reaction time, the PFOS concentration started to decrease, and we created inorganic products like fluoride and sulfate in the system. And all transformed PFOS was converted to fluoride and sulfate. And then we also looked at the effect of the structure of this different PFAS, looking at C4, C6, and C8, and we found that we need higher contact times for shorter chains, which are even more persistent, and also the transformation magnitude was greater for the substances that had sulfonate group compared to those that had carboxylate groups in them. And also we looked at the effect of temperature, and we found higher transformation to happen at 50 degrees C, and we did not see any transformation at 40 degrees C. And again, we supported the previous results where the transformed PFAS was all converted to inorganics like fluoride and sulfate. And also we were able to simulate the nature of whatever happens in groundwater by conducting column experiments where we pumped PFAS through a column which was packed with sand and nanoparticles, and we were able to transform 58% of introduced PFAS. So given this information, I would like to have a summary that, and also to emphasize that PFAS does occur in organic waste products, and the total loads were similar for biosolid based products and also the organic fraction of municipal solid waste, and it was around 30 to 80 microgram per kilogram. And we had exception for one biosolid based product which has almost 185 microgram per kilogram. And the precursors that we found in these products were similar, and they could lead to PFAA generation. And when we investigated the remediation of PFAS with the technique we used, we found that fluoride and sulfate are major products of PFAS transformation, and shorter chain PFAS need longer contact time to be transformed. Temperature affects PFAS transformation, but interestingly, we didn't see the highest transformation at 60, but at 50 degrees C. And also the results that we found, almost 60% of PFAS is removed through the system we have simulated. It's encouraging to do more experiments and investigate this process. And also there are more permeable reactive barrier column studies ongoing in our research group now. And by this, I would like to end my talk, and I would like to acknowledge my colleagues who have done this research and have helped me to give these results. Thank you.
Hello?
Sorry about that. I had my own mute button on. This is Rebecca. And thank you, Masa, so much for your presentation. And given Masa's acknowledgments, I want to also acknowledge Dr. Lois Wilson at Michigan State University, who was instrumental in helping us put this together. And thanks also to Ann Nardi, who's working in the background and helping us with our social media, handles marketing communications for the North Central Region Water Network, for her role in putting this together. So with that, we'd like to introduce Cheryl Murphy, who is an associate professor of ecotoxicology of fish, ecotoxicology of fish at Michigan State University. And Cheryl is going to help us understand. Okay, so what does all of this mean in terms of our own risk assessment, both ecological and agricultural risk assessment? So thank you, Cheryl, and take it away.
Thank you very much. Thank you for having us. And this has been really informative. So yeah, that's where I work more in the natural resources world, and so concerned with how PFAS is moving through all of these different matrices to get into natural resources and agricultural products that eventually lead to human exposure. And so that's where we're mostly focused on here. And I mean, it's a big concern. There's a lot of different, like, do not eat advisories. We have them in, there's just an example from the Michigan DNR, it's Fish and Wildlife Consumption Advisory Committees. They have all these do not eat advisories for fish that have any levels of PFAS in them, in the Au Sable River, for an example. And so, I mean, and the problem is that we have lots of fish species, lots of wildlife species out there, and we're starting to detect all of these chemicals in them, and we have to try to figure out what is a safe eating level, safe exposure level. We're also finding out that this just recently near Oscoda, they found PFAS in deer with really high levels of PFAS concentrations in one deer, and so had to put out a do not eat advisory for PFAS. And so it's kind of unusual, and we don't usually see these types of things in deer. So so there's a lot that we have to sort of explore for PFAS. What we don't really know, there's a lot of people working on these issues now, but we really need to have a good handle on things like exposure pathways, how PFAS is biomagnifying in the food web, what are the biological effects on all these different taxa. We just have to have a good understanding of risk, and it's challenging. What we do have to then is there's a lot of things that we have to do. We have to figure out the pathways for exposure. How is PFAS moving through the environment, through our ecosystem, into our natural and agricultural products, and then what are the biological effects? Because some of these, there's over 5,000. There's about 5,000 of these chemicals, and not all of them are going to be toxic, and some are going to be more toxic than others. And we actually have to figure out which ones are going to actually elicit some sort of biological effect. And it's where they intersect, how exposure and biological effects sort of intersect. That's when we start to characterize the risk for these things. And so basically, I mean, we could have, this is an example, so we could have a lot of exposure, but if it's not intersecting with the biology, there won't be, and there's no sort of combination. There's a pointer here. Can't get the combination, then there's no risk. But if the biology and the exposure is intersecting, then those are things that we have to be really concerned about. So how do we do this? Well, we have to figure out how these chemicals are bioaccumulating, bioconcentrating, and biomagnifying, and all of this is uncertain. And these chemicals have kind of unusual properties, and they don't have definitive. It's not like a PCB where we had 209 of them, and we could characterize them pretty carefully. These ones the properties are all over the place. And some of the properties, for example, they bind to proteins like albumin and also the membrane phospholipids. So in the cell membranes, they're going to be binding to those membranes instead of the storage lipids. And we're used to mostly the stuff that we characterize, we're used to chemicals that are binding to storage lipids rather than these types of chemicals. A lot of them can be metabolized, but we don't know what the rates and the mechanisms are. A lot of the next generation of PFAS have not been
Cheryl, I have lost your audio. I don't know if others. Okay, thanks for that confirmation, everyone. So Cheryl, we have lost your audio, and I'm assuming Brian, our man behind the scenes, is helping us figure out what's happened with Cheryl's audio. And while we're doing that, I know Cheryl's just pretty much gotten into her presentation, but just a reminder to everyone that if you have questions on our previous presentation or the start of Cheryl's, you can go ahead and submit those in the chat box. Folks are doing that well. Oh, Cheryl's back. You can hear her audio. Cheryl, are you there? Okay, while we wait to get confirmation from Cheryl, I'm going to go back and start with questions, and we'll just have to ask Cheryl to chime in if she has an answer that doesn't get covered by our first two presenters. So Kelly, or wait, I think Mark had one before Kelly. Yeah, okay, Mark asks, are local water tests for city and county bodies of water available, and what agencies or offices would be doing those tests? So Courtney or Amasa, maybe you could answer that, and Cheryl, when feel free to chime in right away.
Can you hear me?
Oh, yes, we can.
Okay, I'm sorry, I don't know what happened there. So.
All right.
All right.
Keep going.
Okay, so we'll just push pause on that question mark, and we'll get back into Cheryl's presentation, and we'll save your question until she's through.
Where did I leave off? Just here with so you guys know that then I don't know where did I okay, that we just don't know how these are bioaccumulating and bioconcentrating. Is that okay? I can move on. So
perfect.
Okay. All right. Sorry about that. So what is needed is we need to figure out some standards for PFAS and identification of unknowns. There's a lot of precursors out there. There's like chemicals, the PFAS precursors out there, and we don't really know if they're all combining out. There's controlled laboratory or semi-field dietary biomagnification studies on fish, mammals, avian, and plants. Those need to be we need to have those done. We need laboratory studies in which fish are exposed to contaminants through their diet, not just by respiratory uptake from water through their gills. And we need more of these types of experiments. We need experiments that expose mammals, such as the deer, the mink, and others to background levels of PFAS in drinking and feed, and we need this in birds. We need to figure out the half-life for all the different PFAS and the trophic structure studies for specific impacted areas, like maybe using some stable isotopes. But basically what we really need to do is develop models, because this is such a complex problem. We're not going to be able to do it with very simple experiments. We're going to have to develop models for contaminant, fate, and transport. And so that's just for exposure, and just to figure out the biological effects from exposures. We usually do these dose response sort of studies. This is the hazard of what ecological risk assessment is, and we need to determine what the hazard is to biological life. And the problem with the PFAS chemicals is a lot of these chemicals are not lethal, but they are sublethal, and so they could be impacting long-term population health because of effects on the reproduction, the growth, and the immune function. And also because these compounds are not occurring in isolation. You're not going to get one single PFAS. You're probably going to get a mixture of PFAS, and they're probably also occurring with other legacy contaminants such as mercury and PCBs. There's going to be combination effects that we're going to try to we have to figure this out. And because the way that the PFAS they bind to things like on the cell membranes, and they interfere with protein functions, they could actually amplify the effects of some of these legacy compounds. All right, so this is where this is I work. Most of my career is in this adverse outcome pathway framework, where what we're trying to do is we try to organize all of the biological impacts along levels of biological organization. So we want to go from like the toxic effects into the molecular level, cellular, organ, and we try to create these linkages between levels of biological organization and causal linkages. And by doing so, that means then you can start to, if we can start to create these linkages between levels of biological organization in a meaningful and a robust way, then we can start to measure things at the cellular in high throughput ways, which is much easier to do than doing whole exposure studies on whole organisms and whole systems. And so the idea is then if we can start to tackle what's happening at the cellular level and infer that how that's going to happen at higher levels of biological organization, we can start to tackle problems, especially mixtures and multiple. Okay, so that's what we need to do. We need to test on molecular and cellular levels of biological organization to determine potential toxicity of pathways. It's relatively inexpensive compared to whole organism exposure studies. We get this mechanistic information that will inform interactions with other contaminants and stressors, and then we need to link to adverse outcomes that can be interpreted at the population level for ecological risk assessments. All right, and so many of these PFAS are being run through this EPA ToxCast system to determine the molecular responses and cellular responses. So they're starting to look at all these high throughput screening to see where these chemicals are hitting. These molecular level effects will have to inform population models calibrated for Michigan fish, wildlife, and agricultural resources through these AOP models. And we will need representative fish, amphibian, avian, and mammalian models, and thousands of these PFAS have to be assessed. But once we set this framework up, it can be used to respond to any future stressor and multiple stressors. And just as an example what an adverse outcome pathway is, this looks a little technical, but basically we know, for example, that it hits the PPAR alpha binding. And so basically if it hits this chemical, hits this receptor, it'll cascade up the levels of biological organization. If we can create all these linkages between these levels of biological organization to eventually get something that, like, it causes protein catabolism, which will then cause like a weight loss. And that is something that we can incorporate into some population models to determine the population effect. And so basically, so that's just I'm just about done here. Basically want to characterize the biological effects. We need to figure out what the pathways of exposure, and then the combination of those are the ones that we want to really hone in on and determine the risk. And I do have a new project that's been funded through the Michigan DNR to look at the fate, transport, and bioaccumulation of PFAS in the Huron River watershed. We have a fate and transport model of PFAS in the river proposed, and we're going to be looking at the toxicokinetic models of PFAS and bluegill. And so I'd like to thank everybody for their help with this. And sorry for losing you for a bit there.
No problem. Thanks so much. We glad we got you back, and thanks for helping to provide some additional context for how how we think about approaching risk with these chemicals. Okay, so let's go ahead and circle back around to our questions. We'll just move to that Q&A session so you can see the names of our presenters there, and we'll go back to Mark's question, which is he's asking about local water tests for city and county bodies of water, and are they available, and what agencies or offices would be most likely, do you think, to have that information? And I'll open that up to all the presenters, whichever you think, whichever of you would like to answer.
I think I have an answer for this question. So I think, yes, they are private companies that would like to do it, but it's not through the state and costs vary. But to confirm it, I mean, for Michigan, I know you can call the Michigan Environmental Assistance Center, I believe. And I'm not quite sure how it goes because the quality assurance, quality check for measurement of PFAS is very important, meaning that we have high contamination levels when we are trying to measure the concentrations of these compounds in different media. So yes, the answer is yes, there are private companies, but I am not quite sure how you can make sure about their performance.
Courtney or Cheryl, anything to add? Okay, I will take that as a no, and we'll go right on to Kelly's question, which is about a sort of specific area in the landscape. Are any of the presenters aware of additional studies being conducted on leaching of PFOAs or PFAS and she says PFAS specifically from turf fields, especially when they are located next to a surface drinking water supply?
Of any studies, but I imagine, I mean, there's a lot of sampling that's being done across water supplies, and I think what happens is if you get a hit for PFAS and people, then they try to figure out what the source of it would be.
Great, thank you. And it seems like, of course, as people's awareness of this issue increases, and by people, it would be municipalities, counties, the federal government, and private companies, and even private well owners, they would be doing some of this sampling on their own. And Kevin has a question about farmers. So he says, I work with farmers who have extra manure storage capacity. They are sometimes asked by dairy processors and others to accept, quote, local non-farm liquid waste. A number of farmers are becoming skeptical of this, which is understandable. And PFAS is one of the reasons they are becoming skeptical. They worry about accumulation in soil and transfer to their animals. Any response to this practice of farmers accepting non-farm liquid waste, or how should farmers go about, making a decision on whether to accept this waste?
Okay, I would like to add that the manure storage, I don't think will have any PFAS since we looked at different products that were only manure. So I think they should be fine in terms of the manure storage and PFAS sources. But honestly, I am concerned about the local non-farm liquid waste because we had some research going on and we found that PFAS can leach to pore water and can be taken by plants, like a plant uptake problem. So I am not sure about the animals because if they are consuming this corpse, I believe it can accumulate in animals and it can be ending up in our food chain. If they are contaminated with PFAS, I mean this, because they need to be checked at non-farm liquid waste to see if they are contaminated with PFAS or not.
Okay, so then, just to follow up on that, then it would the answer for farmers to be to request that there's some testing before they accept non-farm liquid waste?
I hope
That-
that this is possible.
Yeah,
Sorry.
that would be ideal, I think, too. Sorry, I didn't mean to interrupt. Yeah, if they could get. But there's not a lot of standards that have been developed for PFAS in all the different types of PFAS and different types of matrices. And so I don't know if the science is there yet, able to do routine testing yet for everything that's out there.
Okay, so ask about testing and then understand that, you know, they may be testing for certain kinds of perfluoroalkyl chemicals, but they may not catch everything.
Maybe they can also ask about the source. So where is this liquid waste is coming from? So that should be an option.
Perfect. Thank you so much. Okay, next question from Mark. Are the use of PFAS ground filters regulated through local governments and their regulations?
I don't know.
I don't know the answer to this question. Sorry, I'm a researcher. I'm not sure of the policies involved in this.
Okay, you've stumped our presenters. If anyone knows the answer to that question and you want to type it into the chat box, go ahead. I know we have a lot of expertise here on the line. All right, Monica Jean, should we be having, quote, do not pump, quote, unquote, referring to irrigation with farmers due to levels being too high in groundwater? So should farmers be cautioned against pumping groundwater that is high in PFAS? And Monica Jean is in Michigan.
Aware of how the rules should work on this. I mean, who can really give this act of do not pump? But if they are aware of how levels of the PFAS in groundwater, I believe they should call this, I mean, Michigan Environmental Assistance Center. I believe they are very active, and Michigan is one of the states that is really active on PFAS, and they are taking care of this. So I believe they need to call this center to get more information about it.
Yeah, the Michigan PFAS Action Response Team. There's a website, and they're testing and being pretty proactive about where PFAS is and sort of the steps. So I think that you're right. That's a good place to start.
If one of you could put those links in the chat box, that would be great, so everybody could see those links. Thank you, Brian. Okay, next question from Mark, a different Mark with a C. Is there, let's see, is there an effort in Michigan and in other states, if you're aware, we saw some maps in this regard, but is there an effort in Michigan to create maps of known impacted water bodies? And if so, are there efforts to notify adjacent landowners and entities using that source as irrigation water? So this is also related to irrigation, but I think the mapping question could apply more broadly.
So again, I think this is with policy. I think so the question is whether or not they notify landowners. I think again you'd have to get in touch with your where is this question based?
This question is from Michigan, so it's, if you look in the chat box on the right hand side, it's right after Monica Jean's question, Mark with the CG number 2, and he says he's in Michigan.
I think that's a question for the Michigan PFAS action team. I think so. So you
Okay.
I would just contact them.
Okay, and Brian has put a couple links there in the chat box, Mark, so you can find those links there. And Noah asks, it seems like this is a very risky class of chemicals with persistent, widespread effects. Should the regulatory, quote, bar for allowing production of new PFAS or PFOS chemicals be higher? Given that there are so many, you know, is there any value in a precautionary approach for new chemicals like this?
So I should say, I believe it was in 2015 that EPA announced 70 ppt for C8 compounds, but unfortunately, companies are producing some other PFAS compounds and they are naming them differently. Let's say like GenX, which is very, very in demand right now. But these are again PFAS, and I'm pretty sure EPA is still working on the regulations, and I think states are also defining their own regulations for the production of these compounds. And I should also add that the PFOS and PFOS, the C8 compounds, were completely phased out in 2015, but it only happened in US. So it's possible that other countries are still producing them and using them.
Okay, thank you, Masa.
I can answer too. I mean, so the question is it's from Noah, right? And they want to, so why aren't we regulating these chemicals better? Up until like recently, we've been just mostly regulating chemicals on acute toxicity, and so these sublethal effects don't get really considered. Recently, in 2016, the Toxic Substance Control Act was modified with the Frank R. Lautenberg Chemical Safety, where they're trying to incorporate all these high throughput testings to try to tackle the sublethal effects. The science is evolving. We're just not quite caught yet, I think, to be able to tackle them all. And if we can get all of these high throughput screening things in and try to pre-screen chemicals before they're released, that's great if we can get there. But the science is slowly is getting there. It's rapidly, this last decade or so, it's moved really fast. But we still have work to do before we can make this into a regulatory realm.
Thank you, Cheryl. That's a helpful addition. And just for everyone, it looks like Courtney's having some problems with her audio. Maybe she'll be back on in a minute here. Okay, she's good now. Great. Courtney, did you have anything else to add?
No. Good answer, Cheryl.
Okay, thank you. Next question from Tim. Is there any research on whether compost made with food waste has higher levels of PFAS than other compost?
Yeah, I think I had this in my slides. I believe if you will have this document later, it should be in slide 9. And the compost that had food waste has much higher concentration of PFAS than other composts. So the answer is yes.
Okay, thank you, Masa. And from Bill, any research on filtering / removing PFAS from contaminated or source environments? I mean, I think we covered some potential options for that, but is there anything else that any of our presenters would like to add?
So I think this is a remediation question, so I would get it. I believe in our research group, we've been doing a lot of research on breaking down these compounds. Unfortunately, I don't think if filtering will remove them. And so far, there are really limited options that they're working for removal of PFAS. And I think the answer is no, there is no filtering option that can really remove PFAS from contaminated environment.
Definitely more work to be done there. Thank you, Masa. Okay, we have some links here that you can see in the chat box provided by our participants and our presenters. And then a question from Lois Wolfson. Is the PFOS being found now in groundwater or surface water a result of the newer chemicals breaking down to PFOS, or are they more legacy?
I think both, because the new compounds can break down to PFAS. We're working on it, and they're breaking down to PFAS. And also, since PFAS is very persistent, and if we are not doing any action on taking them out, so they will stay in groundwater or either in surface water. So both of them can end up to PFAS concentration in surface and groundwaters.
Great, thanks. So there's some complication there in terms of understanding whether the source is more recent or whether it's a result of breakdown from previous contamination, it sounds like. Yeah.
Yes,
okay.
exactly.
Thank you. Another question from Kevin Klingberg. He says, this is something I received from Wisconsin DNR when he asked about soil and materials testing. There are labs that will provide soil and liquid material analyses for a suite of PFAS chemicals. However, there's no promulgated EPA method for any media other than drinking water. Labs may refer to soil and liquid materials PFAS analyses as PFAS in solids and PFAS in aqueous non-drinking water, respectively. So just a little bit of additional guidance from Kevin from Wisconsin, and then he also lists a link to EPA-approved laboratories. So thank you so much for that, Kevin. And I see there's maybe some additional typing going on here. If you have one more question, I think we have time for one more question, so I'll just wait for a minute here and see if that comes through. Okay, and if not, oh, yeah, or Mark just has a comment about when will our society adopt the precautionary principle? Well said. And just thank you so much to our presenters and to our participants. Sometimes in these webinars, we get used to just sitting and multitasking, but we really appreciate the time of our presenters and the engagement of our participants. What the North Central Region Water Network is all about is working on these issues together. And thanks again to Lois Wolfson for helping us put this together, and to Ann Nardi, and to Brian Adams, and to Martha Martin for all the assistance. You can visit northcentralwater.org to access this recording and also our archive of many other different water related webinars. So thank you so much, and thanks again to our presenters for joining us today with this amazing information, and have a great day, everybody.